So first of all, what is a biofuel?
A biofuel is a fuel that is derived from a living thing. So it's going to be from plants or something that is fermented by a microbe such as yeast.
Now, I'm assuming we go to biofuels because we think they'll be either energy efficient or help the environment. Is that right?
Yes, well, mostly it's towards helping the environment. We really want to get away from the use of fossil fuels.
But biofuels, if they're from something like corn, for example, you still got to grow the corn.
Yes.
You got to farm it. It requires lots of land and water.
Yes.
Growing that product corn is energy intensive in itself, is it not?
Yeah, so currently there are some biofuels that are out there. So we call those first generation biofuels, and that is derived from corn so that you get the sugars from the corn and you feed it to yeast and they ferment it to ethanol. Or you can get biodiesel from oil plants such as soybean. The problem with that is those are on agricultural land that we compete with for our food. And so really we're moving towards what is termed second generation biofuels, so things from different wastes or from plants that can grow in areas where we can't farm. So areas like the Pacific Northwest where it's really dry and arid. So we're looking at being able to utilize those products as feedstocks for the microbes to convert to biofuels.
How do microbes work on these products to give us biofuels?
So in terms of some of those plants, you can't directly feed that to a lot of microbes. But another way you can get at that is you can take the enzymes from those organisms that can break those down into sugars and treat that biomass with it and the converting it to sugars. And then you can feed it to a conventional yeast. It's pretty intensive and it's not cheap. And that's why you really don't see that yet.
So tell me a little bit about the research you do in biofuels.
We're working on non-model microbes. So yeast is more of a model microbe. It's been utilized for centuries for the production of ethanol. We've used it to study things such as cancer. There's a lot of genetic tools available. But when you're moving to more non-conventional microbes that have unique capabilities, they don't have those same genetic tools available. So we spend a lot of time here at Oak Ridge developing new genetic tools for these organisms so we can engineer them to make the products that we want.
So speaking of new genetic tools, there's all this buzz around CRISPR. Tell us, what is CRISPR? How does it work? And is that something that you're using?
Yes. So CRISPR is actually an immune system for bacteria. So it was just...
Time out. You just said an immune system for bacteria. Help break that down, because that's really exciting. Bacteria actually have their own immune system?
Yes. So there are viruses out there called bacteriophages that directly infect bacteria, and that can cause issues for the bacteria.
Sure.
So the bacteria generated these Cas systems, so CRISPR-Cas, and so they basically can identify foreign DNA, cut the DNA, and it'll get rid of it and clear it.
That bacteria can use the scissors and cut out that foreign virus DNA and save themselves.
Yes.
Got it. Okay.
So we're actually using CRISPR to modify bacteria and yeast in our lab. It will cut the DNA on both strands, and that usually results in a mutation. And so an insertion, a deletion of a few base pairs. So a lot of times when we're doing science, we want to look at deletion of particular genes to better understand what its function is. So when it's not there anymore, what happens? What's the phenotype?
So you're using this great technology, CRISPR, essentially genetic scissors that can cut out DNA. And what you're doing is you're using it to cut out DNA to either remove genes or use it to insert certain genes that you want. What are you trying to select in your genetically modified bacteria and yeast?
So linking a genetic change to an output phenotype. And so in terms of selections, what we can do is if you're making a very toxic compound, but you want them to be able to still grow, and you can select for things that can still grow under those conditions and look at what genetic changes allowed for them to grow better. We've been building very large CRISPR libraries where we can target all over the bacterial genomes.
You're figuring out this whole library of CRISPR tools that can really be useful on all these genetic modification possibilities. With all of that, 5 to 10 years from now, what do we expect in biofuels?
I think in 5 to 10 years, we should start to see things that are starting to come out of this space where they'll be usable. We are moving towards technologies where we can take that ethanol and we can use catalysts with the engineers and convert that to sustainable aviation fuels.
So that's so interesting. Dr. Eckert, you've modified millions of trillions of bacteria by this point in your career. Do you have any examples of how they react after you've modified them?
One great way we can do that is doing the CRISPR interference in those cells, and then we can look in real time. It's basically taking like 5-minute snippets of the population over time. So you're seeing cell division in real time.
So in real time, we're watching bacterial activity as a consequence of the modification you've done.
Yeah, so one of which will be the wild types. It's unedited, so we don't have any changes to it. It's what it is in nature.
So right now we're looking at the regular non-modified bacteria.
Yes. Yeah, and so you'll see it dividing evenly, and so all of the cells are similar sizes as it divides. But in the case of this other video, we also have CRISPR interference happening, and it's targeting a gene called FTSZ that is responsible for basically cell division. If you knock it down, these cells can no longer divide and they just continue to elongate.
So it's almost like a string-like series
Yes,
of
it's
bacteria.
basically like a snake.
This is really exciting. I look forward to following your research.
Thank you.